Hello, and thanks for joining us today for another TissueFAXS webinar. My name is Carly Briffa, and today we're joined by our guests, Dr. Andrea Papadopoulos and Trevor Khaba from the Ndhlovu Lab Group and CESORA at the African Health Research Institute to present their research on harnessing imaging technologies to expose hidden HIV reservoirs.
Thank you, Carly, for the introduction. So today, Trevor and I will be talking about harnessing imaging technologies to expose hidden HIV reservoirs. There are currently 39 million people around the world living with HIV. Unfortunately, 29.8 million are receiving life-saving ART treatment, making this a manageable chronic condition. But we are still seeing new infections every year, and people are still passing away from illnesses related to acquired immune deficiency syndrome, which is when the body runs out of CD4 cells. And here in South Africa, where we're presenting from, this is the country that is the most affected by HIV in the world. To know how well people are living with HIV, their viral load is monitored, as this is a measure of how well ART treatment is suppressing the virus. And what this bar graph shows is individuals who are not reaching viral suppression. And while the situation is improving, you can appreciate that not everybody receiving ARTs is suppressed, and that's the black bars. Not everybody who knows their status is receiving ART treatment, and that's the dark gray bars. And not everybody knows their status in the first place to become part of the HIV care continuum. And so these individuals who do not have viral load suppression are also able to transmit the virus, and that perpetuates the cycle of persistence of HIV in the human population. One of the reasons why a person might not achieve viral suppression is the persistence of HIV viral reservoirs. And this is the greatest barrier to curing HIV, as these viral reservoirs, which are cells infected with HIV that cannot be cleared by ARTs or the host immune system, persist within the body throughout the course of infection. They are mainly found in tissues in the body that are not accessible by ARTs, and they're a source of viral rebound or replication at the point of treatment failure or if the treatment is interrupted. And that's what makes ART treatment lifelong. The key to curing HIV is understanding HIV reservoir biology, and we do this in KwaZulu-Natal, looking at two main clinical cohorts here in this province. So the first study aims to couple social interventions with basic science research. Enrolling women who are initially HIV uninfected, the women visit the site bi-weekly for HIV screening and economic empowerment classes and activities. If they do acquire HIV, they are initiated on ART within 3 days of testing positive. And this enables studying HIV reservoir development in the context of acute infection and early treatment. And because most reservoirs occur in the tissues, this study overlaps with the UKZN lymph node study, where we collect lymph node excisions from participants. So our lab explores these lymph node and blood samples to characterize the HIV immune microenvironments. So we do this looking at HIV-specific T cell responses, macrophage infection, studying post-mortem tissues, and doing in-depth HLA typing across the continent. We use multiple approaches, including multi-spectral imaging, high-dimensional FAXS, single-cell transcriptomics, and we've recently set up the Center of Excellence for Spatial Omics in Africa, where we do spatial omics and next-gen sequencing, among other state-of-the-art technologies, to answer important biological questions here in the continent of Africa. So my next slides will showcase some of the research that we're doing, and my colleague Trevor Khaba will go more in-depth into the multi-spectral imaging work that we do. So to study HIV reservoirs, we need to be able to detect the virus directly in tissues. So using high sensitivity imaging, the P24 gag protein can be detected in lymph nodes, even in individuals who start ART really early after infection and who go on to achieve full viral load suppression. We can detect active virus by imaging RNA using RNA-scope, and we can also measure proviral DNA using DNA-scope imaging. And when we quantify the HIV in these tissues, we notice that individuals who start treatments really early after infection drastically limits the amounts of HIV viral reservoirs that can be established in the body, whereas the longer HIV goes untreated, the more reservoirs can be established. We also found that HIV viral reservoirs are localized specifically to B-cell germinal centers. And these are tightly regulated antibody factories. And this is important because when you look at the blood, the host immune system can actually assist ARTs to suppress the HIV viral load by using these CD8 cytotoxic T cells, which are able to kill HIV-infected cells. But this is likely not the case in these germinal center antibody factories, where these antigen-presenting cells need to be preserved. And these antigen-presenting cells are also the target cells of HIV. And so when we image CD8 B-cell markers and the P24 HIV gag protein, we can see that these CD8s are largely locked out of these germinal centers. But because these imaging technologies are so sensitive, we can still detect a tiny proportion of CD8s that are able to infiltrate these germinal centers. And so even though there is a limited amount of CD8s in the germinal centers, we still don't see clearance of HIV. So to better understand this phenomenon, we are currently using image analyses to characterize the HIV reservoir immune microenvironment. So using serially stained lymph nodes for cytolytic markers, for example, and measuring the distance of these cytolytic cells from the P24 protein, we can confirm that cytolytic cells lack access to HIV reservoir sites, and CD8 cells that do occur inside the germinal center lack cytolytic responses. So to get a view of what is governing these processes, we are using the highly sensitive imaging of P24 in combination with spatial omics to be able to determine how immune gene expression changes in the presence of HIV infection. And we can see when we cluster these immune gene expression patterns that these P24 positive or P24 negative germinal centers form clearly separate clusters of immune gene expression, indicating that HIV really modifies the germinal center immune microenvironment. So in future, we can validate some of these genes of interest using multi-spectral imaging. And so now I'll hand over to Trevor Khaba to talk more about the imaging that we do.
Okay. So expanding our research repertoire to include immunohistochemical staining was born from the need to find multiple approaches at answering the questions on the HIV reservoir that could not be addressed solely using cytometric methods. IHC staining provides us with spatial information within the natural context of the tissue. It allows us to visualize protein expression in relation to cellular and tissue structures. It allows us to study the dynamics of tissue immune responses within the framework of normal tissue architecture, and it adds a different and important dimension to our research. So in 2018, we purchased an Axial Observer imaging system from TissueFAXS. We had selected this system for a number of reasons. The system allowed us to acquire entire tissue sections at a much higher resolution and speed than the scanner we previously had access to. The momentum in our data generation naturally pushed our lab to upgrade the system. We upgraded the microscope to an Axial scanner with 8 custom filter sets. These additional filter sets meant that we could extrapolate much more information from each tissue section in order to address many more difficult questions that we are faced with in our research. The system also provided a very robust imaging analysis software. However, with our upgrade, we have also upgraded from the TissueFAXS single-cell TissueQuest analysis software to their more advanced StrataQuest analysis software that is more geared towards studying tissue compartments and their spatial relationships. This is much more complementary to the spatial neighborhood analysis approaches that we are interested in our research. So since we purchased the imaging system, we have had a number of successful publications that have utilized the system and still have many more in the pipeline. So one of the main areas of interest in our lab has been looking at HIV reservoirs. We have been able to publish data suggesting that despite the initiation of early antiretroviral therapy in HIV infection, the virus continues to persist in germinal centers within lymph node follicles. And in fact, there continues to be active ongoing replication in these sites, despite successful ART therapy, which poses an obvious challenge in achieving complete viral elimination. So a possible contributing factor to the persistence of HIV in the germinal center is the spatial segregation of CD8 T cells that are the primary adaptive cells tasked with eliminating infected cells in the immune system. Understanding why the spatial segregation exists might help in understanding these sites and how to disrupt them. Moreover, we observed that despite the spatial segregation, there is recruitment of CD8 T cells to these sites in response to viral protein. However, these follicular CD8 T cells have an attenuated cytolytic ability, and from data generated from our spatial omics multidimensional analyses, we believe that they might have an altered phenotype that is geared more towards regulatory and anti-inflammatory activities that further promote the persistence of virus in these sites. So we are trying multiple approaches in our investigations, and they are leading us down many interesting paths of inquiry and hopefully towards meaningful answers. We are also happy to report that our lab, with the help of the Bill and Melinda Gates Foundation, have started the Center of Excellence for Spatial Omics Research in Africa, also known as CESORA. CESORA provides a range of education and training opportunities to budding researchers through hands-on training and project-based learning. These educational and training opportunities are aimed at equipping researchers with the necessary knowledge and skills to use, develop, and apply spatial omics technologies to studying diseases in the native tissue environments. This year, CESORA hosted its very first intern from Harvard University, who spent 9 weeks working extensively with CESORA's geomics technology to better understand the transcriptional profile of HIV-infected macrophages within lymph node tissues. CESORA has also hosted its first annual in-person theory and practical workshop that covered the entire spatial omics research workflow. For more information on CESORA, you can visit the link on the screen. These are our references. And I would just like to acknowledge the program and lab PI and director of CESORA, Professor Ndhlovu, for his leadership and mentorship. I would like to acknowledge our Ndhlovu lab team, including our collaborators, who work tirelessly to ensure that we are continuously generating and publishing data to contribute to the collective knowledge pool. I'd like to acknowledge the study participants and staff involved in providing us with the invaluable tissue samples that are at the core of the work that we do. I would like to acknowledge the Bill and Melinda Gates Foundation for funding CESORA. I'd like to acknowledge the Reagan Institute for funding the purchase of our imaging system. I would also like to acknowledge all of the other institutions that have contributed to making all of this possible: the NIH, HHMI, HPP, RE, Sante, UKZN. Thank you.